Hi everyone, my name is Rebecca Sempt. I'm a postdoc in Beth Simony's lab in the imaging platform at the Broad Institute of MIT. Today I'm going to be giving you a tutorial about Cell Profiler Analyst, which is a software our lab has built to help you visualize and explore the data that you generate with Cell Profiler. So I hope that you come away from the video today with a greater understanding of how to use Cell Profiler Analyst and a greater appreciation for how to turn the numbers that you get from Cell Profiler into biological insights. All right, so let's get into it. As a roadmap for today's video, I'll go through a brief outline first. I'll orient you to the software, and I'll give demos throughout to explain how to use different modules and different functionalities within Cell Profiler Analyst. I'll introduce you to the translocation dataset, which is the dataset we'll be using for the tutorial today. If you've been to some of our other Cell Profiler tutorials, you may be familiar with this dataset already. We'll also talk about using the classifier to train a machine learning model to classify your cells into different phenotypes. You can train the computer to determine which features in your data are most important for classifying cells into two groups. And then you can use that output, either just export it and look at the data, or you can put that back in Cell Profiler and use this as a module that will classify future data sets using those same rules if you'd like. As I just said, we'll also talk about exporting the classifier to use back in Cell Profiler. And finally, we will touch a little bit on 3D data and some of the recent improvements that have been made to the classifier tool to allow you to classify objects from 3D data sets. All right, and with that, we'll get started. This tutorial is aimed at people who are really kind of beginners, who are maybe opening Cell Profiler Analyst for the first time. I'll demo things as I go along. Feel free to, since this is a video, pause, go back, watch it again. And hopefully, like I said, you'll come away with a greater understanding of how to use the software and how to transform numbers into biological insights. All right, so with that, let's get started and take a look at the main window when you open up Cell Profiler Analyst. So this is what it looks like. And again, I'll do a demo in just a moment, but there are a couple of things just to point out first. Here in black is a log that will print as you use the software. So if you have errors or you encounter difficulties or just want to know what's happening, this is a good place to look to understand what's happening in the software. Up at the top, we have various icons that will indicate different functionalities we can use in the software, like the image gallery, which will allow us to view individual images from our data set and objects from our data set. We have the classifier, which we'll spend a lot of time on in the future. The plate viewer, which can allow you to get a bird's eye view of your data set as it relates to a plate well format, and various graphing tools. We have scatter plots, histograms, etc., and a table viewer that will allow you to view the measurements directly that have been created by Cell Profiler. These are the main functionalities that we have within Cell Profiler Analyst. However, the first thing that you'll notice when you launch Cell Profiler Analyst is that it will prompt you for a properties file. Now, a properties file is really just a text file that has some configuration details about things like where are the images located, where is the data located, where is the database located. So this can be output very easily using Cell Profiler's export to database module. So before we've been showing you Cell Profiler Analyst, I just want to show you how to do this, how to export a properties file. If we take a look at this is a window showing the export to database module. Here are the settings in Cell Profiler. So all you have to do to get a properties file is to tick yes here under create a Cell Profiler Analyst properties file. That's it. And then this will create the properties file, and then you can look at your data in Cell Profiler Analyst. There are also some other options here, like which objects do you want to use for your locations. Also, if you have a plate, like the example data today was done on a 96 well plate, you'd include information about the plate type and metadata for the plate and well, which can be informative for like the plate viewer and some of the other views in Cell Profiler Analyst. Okay, so now that we have the real basics of just opening the software down, let's talk a little bit about the biological data set for today's tutorial. Like I said, if you've gone to a Cell Profiler tutorial before, you might be familiar with this one. It's our translocation dataset. You can download this dataset going here, the CZI transloc, bro.io/CZI transloc. The hypothesis here is that treatment with this drug will cause translocation of a marker, in this case GFP, from the cytoplasm to the nucleus. All right, so we can see an example of a non-translocated state on the left here. GFP is in green, DNA is in magenta. You can see the cytoplasm is green and the nucleus of each cell is magenta. The hypothesis is that with treatment of this drug, the GFP moves from the cytoplasm into the nucleus. And then what we see is we don't really see the GFP in the cytoplasm anymore. Instead, we see these white nuclei. They're white because the overlap of magenta and green becomes white. So here's one example of a translocation. This is our hypothesis that the drug treatment causes this. Within this data set, we have the treatment with this drug at a variety of different doses. We also have positive controls for what translocation looks like and negative controls for cells that are definitely not translocated. So that's the brief overview of the data set. As I said, you can follow along and download this data set, the pipelines, everything else at this short link. Okay. So let's get an overview of our data using some of the useful tools for this, the image gallery and the plate viewer. So first, let's take a look at the image gallery, which is this first option here. So here we have our Cell Profiler Analyst window. I've already loaded the properties file, but if I wanted to load it again, I could go File, Load Properties File. We will hit OK. And in this case, I'm selecting the Translocation Tutorial Properties file. We can open that. And now let's use the image gallery just to take a look at a couple of our images and to load up some of our objects as well. So initially you'll get this window that has just two black boxes, one up here for the image gallery and one where objects within an image might be. We can hit this Fetch button to fetch images from our experiment. And we can change this if we wanted to have a different filter besides just looking at the experiment generally. All right, they're kind of hard to see though, so let's go over some of the image controls so we can get a better sense of what these images look like. So up here at the top of the window, we have our channels. In this experiment, we have DNA and GFP. So GFP is in green, that makes sense to me. For DNA, the default will be red in this case, but let's say I want to make it magenta so it's a little easier to see the contrast with the green. Still kind of hard to see. So let's go under View and Image Controls, or Shift-Command-I. This pops up a window of image controls where we can increase the brightness with this slider that has a little sun next to it. We can also increase the size of the individual thumbnails, make them a little bit easier to see. If we wanted to, we could change the contrast stretch to be a log transform, but for this data set, I think it makes more sense to stick with linear. All right, so here are our images. If we want to get a better view of each one, because these are kind of low resolution thumbnails, we can just double click on one and open up a bigger view. Here we could also have the individual controls for, again, the brightness and the zoom level on the image, which can be useful. We can also load up the objects from a particular image by right-clicking that image and then going down here to fetch all objects from image. Takes a second, but that will populate down below all of the cells from that image. So there are 142 cells within this field of view. Sorry about that. If you want to make this a little bit bigger, we can view each of these cells in context by double-clicking it, and that will pop open a window of the original image, where up at the top here we can see this little square indicates where our cell of interest is, so we can see what the other cells around it look like and just get a greater contextual view of what the field of view is. We could also load a particular image if we wanted to, change the number of images that we wanted to fetch, but this is the basics. This is really useful if you want to just look at some random images from your data set, look at objects from those images, get a basic view of your experiment. All right, so let's go back to our slideshow. So in that demo, we talked about how to change colors, how to change the size of tiles and the brightness and contrast, how to, and not listed here, but we also showed how to look at individual cells or objects from each image. So that's the image gallery. Another useful view, especially for experiments where you have multi-well plate format, is the plate viewer, which is shown here. This is really useful for a bird's eye view of the entire experiment and allows us to see, even if we want to, a heat map of different measurements matched to each well where the images came from, which can be a great way of seeing overall, has anything really weird happened in certain wells? Does my experiment look like it is matching expectations on a really, really zoomed out level? So let's take a quick look at this tool. So here's the plate viewer. If I click this, it's going to load up this view. And in this case, we're not using all of the wells. We are really only using this first column, this last column, and this middle row. The first measurement that we have plotted isn't a particularly useful one. It's image number, but we can look up any measurement that we've got here for the images on an image level. We could also look on a per object level if that was useful, or load up a different table, but per image is fine for now. Let's take a look at a different, more useful measurement. So we can load up something like the metadata that we have for whether we are looking at positive, negative controls or experimental data. In this case, these are actually not going to show up necessarily as colors, but what you can see here is we have zeros. These are negative controls in these wells where there should be no translocation. And down here, also negative controls. We have positive controls in the top part of column 12, or yes, top rows of column 12, and in the bottom rows of column 1. So these are indicated with a 1. So those are positive controls for translocation. And then in the middle, we can see this middle row is used for our actual experimental data. Another way of viewing this data would be to look at the dose. So let's go change this to the metadata dose. So here again, we can see the negative controls in blue, here and here, positive controls in red, up here and here. And then in the middle, we can see this transition from a darker blue to more green color, indicating an increasing dose as we move from left to right. Each well has slightly higher concentration of the drug. This is a good way of visualizing what did we actually do in this experiment. And of course, that's not all we want to do. Let's take a look at some actual interesting measurements made by Cell Profiler. If we go down here again, we can select something like, let's look at the first one here, this correlation of the DNA with the GFP. And let's look at it in a particular compartment. Let's look in the nuclei for the correlation with the DNA and the GFP. So this is saying within the nucleus, let's look at the correlation between the DNA channel and the GFP channel. So we might predict that if GFP is translocating to the nucleus, these will become more correlated. So indeed, this makes sense. I mean, we're seeing high correlation with our positive controls up here and up here, down here and up here, and low correlation with our negative controls over here and over here. And then we can see that as the dose increases, we have increasing correlation of DNA with GFP in the nucleus. So this makes sense. I mean, this is some evidence that the drug is doing what we think it's doing. As dose is increasing, the correlation between DNA and GFP is increasing in the nucleus. So returning to the slideshow, now that we've explored the data on a zoomed out level, let's take a look at our specific biological question that we want to answer. And we talked about this a little bit before, but just to reiterate this again, our question is, how does treatment with this drug affect the cellular localization of GFP? Another way of saying this is, how does treatment affect translocation of GFP to the nucleus? And our hypothesis is that the drugs will move GFP from the cytoplasm to the nucleus. And that's kind of what we see happening when we look at measurements in the plate view. So what we could do if we wanted to classify cells into translocated and non-translocated is to pick some measurements. We could set up a filter and say any cells with a measurement above X are translocated and below X are non-translocated. We could include multiple measurements if we want. The trick with this is really that this is kind of tedious and it's not obvious which measurements we should pick. Multiple measurements might be useful, and this might require a lot of trial and error to get right. It's also particularly hard if you have any variation across the images in your data set. So let's train a computer to do this for us instead. So we can have a computer look at all of the measurements that we have available in our data set and select which ones are the most useful in making a classification that we train it on. So we can tell the computer for a subset of data, that is the training set, which cells are translocated and which ones aren't. And then we can train it and ask it which measurements are the most useful for determining this split. And then we can make a model based off of that and apply that to future data sets, or just in general apply it to all of the images in our data set without having to individually classify each cell, which would be super tedious and annoying. All right, so that's the question. That's what we're going to do with the classification, classify objects module. And in this experiment, we actually really have 3 classes of cells. So there's the non-translocated, which we talked about before. These cells have a magenta nucleus and GFP in the outside. The GFP has not moved to the nucleus. We also have the translocated cells. And in these cells, we don't really see the cytoplasm at all. The GFP has moved into the nucleus, which will appear white because of the overlap again, in this case of magenta and green. There's a third type of cell that you might have seen in some of the example images, and these cells are just magenta. These are cells where there is no marker. They are unlabeled cells. They don't have a GFP marker in the cytoplasm or the nucleus. And so we want to take those into account as well. All right. So with that, let's go into looking at the classifier. If we open the classifier tool, we see a couple of panels here. On the top, we have a panel where images will load when we hit this fetch button. We can hit fetch and fetch 20 images, each of individual objects from our data set. We can do the same thing we did before to change the way they look. So I want DNA to be magenta. We could change the channel. So if we wanted to turn DNA off, or just look at DNA, sorry, or just look at GFP, we could do that here. We can also change the colors. I want this to be green and I want this to be magenta. Perfect. And then we can also look at the image controls, just as we did before. Pops up our controller, so I want them to be brighter, and let's say I want them to be a little bit bigger so they're easier to see. So those are our cells. These are currently unclassified cells. Down here we have the classes that we want to put them into. We can do this by dragging and dropping the images. We can also select an image and press 1 to move that into this category, or select 1 and press 2, and that would move it into this category, the second category. If we change our mind, we can drag the image back out into the unclassified bin. It's very drag and drop friendly. We can also add a new class. Down here, this button will add a new one. Let's say we want to call this no marker to represent that third class that we have. And let's say I want to rename some of these other classes. I can right-click the bin, say rename class. Let's call this non-translocated. Oops. And let's rename this other class as well and call it translocated. All right, so let's classify some cells. This cell is non-translocated. We can see the GFP around the magenta nucleus. We can just drag and drop them or press that 2 button to select some cells that are non-translocated. This cell doesn't have any marker, it's just a magenta nucleus. We can put that over here. Do that for some of those cells. And then we've got a couple that are translocated here. Great. And if you're unsure about some of your cells, we would recommend not classifying them, leaving them in the unclassified category for now. So we can fetch more images and repeat this process, adding more of these cells to the different categories. I want to fetch more. And you can do this until you have, say you can start with like 20 or so in each category. Part of this will depend on the number of categories that you're looking at, the complexity of the phenotype, as well as a little bit of trial and error as well. All right, and by movie magic, we now have 20 cells in each of these groups, translocated, non-translocated, and no marker examples. We can then train our model by hitting this train button. Again, we'll see some features here. We could change the top number of features that are displayed if we wanted to look at more of these. We also have a couple of other options over here that I want to highlight. We can evaluate the model, score all of our images, or score an individual image. So first, let's take a look at evaluating the model. If we hit evaluate, it takes a second, this is going to pop up a confusion matrix which will show us how well our particular set of rules performs on our training data. So it's important that the training data is reflective of the overall variation in the overall data set. This doesn't say anything about how well the model will perform on data that it hasn't seen yet. It doesn't say anything about how well it will perform on new data. This is really a measure of internal consistency. We can see on this axis, on the y-axis, we see the true label, and that true label is really just the label I have given the data, the null marker non-translocated or translocated label. And then on this axis, the x-axis, we see, based on the specific rules the computer has come up with, how would it classify those objects? Without seeing the label I've given it, just based on the rules that it's come up with. And so this is really a measure of internal consistency. Is the model consistent with the training set? And what we see here is pretty good consistency. We can see there's a perfect consistency for the translocated. So every single case of truly translocated cell was predicted as being translocated. And then for our non-translocated, it's pretty good, 95%. It's a little hard to see, sorry, it's black on dark blue. And for no marker, we're also pretty good at 95% accuracy. The overall accuracy here is 96%. Ideally, we would want to see this above 95%. This is going to depend a little bit on the complexity of your phenotype, the number of classes you're looking at, and the number of examples of those classes. In biological data sets, it's not totally uncommon to have some classes be much rarer than others, and this can make classification kind of difficult. But ideally, we want to see high accuracy for all of our class types. And this is a pretty good example of this. The other thing I wanted to show you briefly before we move on is correcting examples of bad classification and how to easily do this. After we've trained a model, we can select Score Image and pick an image number, like say image number 1. This will score that particular image and bring up a view where we can see, resize this window, where we can see how the model would score every cell within this image. I find that this is a good way of correcting individual cases of where things have gone wrong. You can also always fetch uncertain examples of cells using the fetch button, but this is another way of doing this. If we hit the controls here, we can see what the classes are. Personally, I find the shades of blue kind of difficult to see, so another useful way of viewing the data is going under View and then hitting View Cell Classes as Numbers. So instead of colors, now we see 1, 2, and 3, and these correspond to no marker, translocated, and non-translocated down here. So if we look at the data and we see an example of a cell that is the wrong class, we can fix that. So let's see, we have, most of these look pretty good, but if we pretend that this was an example of actually this is a non-translocated cell, not a no marker, it should be 3 instead of 1. What we can do is move this window a little bit. We can take that and just drag it into the non-translocated bin and it will pop into the bin. This is a very easy way of correcting things that are wrong in individual images and allows you to do so while seeing a bunch of cells at once. That's another good way to look at the data and to correct the model. All right. After you're done and you're happy with your model and you've evaluated your model, everything looks good, you can save the model by going to File, Save Classifier Model. You can also save the training set this way as a CSV, which allows you to save your work and look at this later. We can save our model. We can also score all images by going to this Score All button. We'll just group by images in this case. We can hit OK. Overwrite our class table. This will take a second. Classify all of the objects in all of our images. We will then pop up a data table that will be our hit table. It will show for every image, for every well, all of our metadata, how many cells there are, and then how many cells there are of each class, as well as probabilities of enrichment for those classes within the wells. This data is now part of the database. We can use this back in Cell Profiler if we want, and I'll be demoing that next. Let's talk a little bit about what you can do once you've trained your model. As I showed you, you can file, export the classifier model. You can also see your training set so you can use this later or train differently in the future if you'd like. These model files that are exported by Cell Profiler Analyst can be used within Cell Profiler. There are a couple of modules that can use these. Classify objects and filter objects are the two. We should note that if you're going to be using the model within any subsequent Cell Profiler pipeline, you need to make sure that the measurements that are used in the model are measured in that pipeline before the classify objects or filter objects modules that actually use the model. So the model needs to have access to those measurements, so they need to be measured earlier in the pipeline. So here is an example window of the Classify Objects module interface. Here are the settings on the right. We can choose the object name that we would like to classify and the location of the model file. An example of this output shown here. We can see we go from all of our cell objects that are measured in our pipeline to being classed as either translocated, non-translocated, or no marker classes. We can see a visual depiction of this classification. In the Cell Profiler output, these classes will also be represented. We can see these as for every individual object within each image, we have a classify class, so which class each cell belongs to in this case, as well as the probability of the model classifying that cell as each class. I wanted to briefly touch on some of the new changes that we've made to Cell Profiler Analyst in order to allow you to classify objects from 3D data sets. So with the latest Cell Profiler Analyst build that is available on GitHub at this link, you can download that, you can install from source. And we can now classify objects that come from 3D data sets. This is an example of one of these types of data sets where you have a Z-stack and cells that are represented that exist within a 3D volume. So in this case, how this works is the plane that is shown for each individual object corresponds to the center of that object. So there's no true 3D view, like you can't slide through the stack and look at individual objects yet, but you have the ability at least to look at the middle plane, which can help you classify objects into different groups. And if you need more help, we have a manual with in-depth details on all of the settings that are possible in SubProfiler Analyst and all of the various tools that we have. You can access that at this link or within SubProfiler Analyst itself under the Help button in the menu. For further help, you're also welcome to make a post at image.sc, which is our forum where people from a variety of open source image software tools post and provide help to users. So there's SubProfiler, SubProfiler Analyst, and many others. So please feel free to post here and we will try to help you with whatever your particular issue is. And with that, I want to make sure that I acknowledge the lab and everyone who's worked on this software. Simini Lab is shown here, and I particularly want to throw a shout out to David Sterling, a previous postdoc in the lab who did a tremendous amount of work on SubProfiler Analyst. I also would like to acknowledge our sister lab, the Carpenter Singh Lab, shown down here, which handle a lot of the profiling within the imaging platform. I'd like to also thank our biology collaborators who provide us with images and with reasons to add more features to SubProfiler Analyst. And also funding from the Center for Open Bioimage Analysis, or COBA, which is shown down here. And with that, I just want to say thank you for your attention. I hope you've learned a little bit about how to use SubProfiler Analyst to analyze your images, to explore your data, and to classify objects within your image data. And with that, thank you. Please check out some of our other tutorials available on YouTube for our other software, as well as other SubProfiler Analyst tutorials. Thanks for listening.